Well, welcome to Club EvMed. It's great to see so many people, and I'm sure we're going to see more tuning in as we go here over the next few minutes. My name is Charlie Nunn. I'm at Duke University, and I run the Triangle Center for Evolutionary Medicine. And TRISEM is one of the main organizers, but not the only organizer for this seminar series. And I'm going to paste into the chat, if I can, the other evolutionary medicine centers that are involved in this effort. And we also work closely with the International Society for Evolution Medicine and Public Health. And again, I'll paste this in to the chat. The chat can't handle all of it at once, so I have to do it in two batches. The International Society for Evolution Medicine and Public Health, as I've pasted in there, and a link to our website. Now is a great time to renew your membership or become a member if you're not already a member. And we always like to encourage people to submit their articles, your best work, to Evolution Medicine and Public Health. And I also want to point out that we have our annual conference coming up in July, at the very end of July, July 28th to 31st. And you're going to see the registration open for that earlier than usual this year. So please keep an eye open for that. Okay, well, I think that kind of covers the intro part I have. I'm actually going to turn things over to Jason Somarelli from Duke University and a colleague of mine in TRISEM, our associate director for the Duke portion of TRISEM. Go ahead, Jason. He's going to do the introduction.
Yeah, thanks, Charlie. Really delighted to see everybody here. Before I introduce Chris to everyone, just our first bit of quick business. We're going to have about a 25 to 30 minute talk here, followed by plenty of time for discussion. So the format of this is we'll hear the talk and then please post your comments and questions in the chat. Hold your questions until the end, and we'll have, as I said, some time for discussion afterward. Not, I'll try to moderate and follow the chat there. I'm really excited to have the opportunity to introduce Chris to the group. Chris McKnight is a senior research fellow at the University of St. Andrews Sea Mammal Research Unit. We were just sort of talking about how this is a world-class place for marine mammalogy and marine biology in general. His research focuses on the diving behavior and physiology of mammals, with a particular focus on seals, as well as freediving humans. Chris completed his undergrad in marine biology at the Queen's University in Belfast before moving to the University of St. Andrews, Scotland, to complete his master's and PhD. He postdoced in the US, France, and Sweden, and then returned to SMRU, where he now leads the ecophysiology lab at SMRU in partnership with Joe Kershaw. And they have some of the best logos you'll see, which are great. Um, his work's been really, an inspiration to our team. And I just wanted to also say that beyond his exciting discoveries and his great science, he's also a fantastic collaborator, and he's one of the most generous and kind-hearted human beings you'll encounter. He's opened his home and his lab both to our trainees, and he's always really willing to hear me out, even when it's a seemingly nonsensical idea. So I really appreciate it, Chris. It's great to have you. Thanks for joining us, and I look forward to the talk.
Thanks very much, Jason. I'll just share my screen here to make sure I get this right. I just double check you can see that okay?
Looks great. Yeah,
That's
thanks.
fine. Well, thanks very much, Jason, for a very kind introduction. I honestly really appreciate it, and just want to, yeah, again, extend sort of my thanks back to you, Jason, both for the invite, but for, I guess, sort of really helping both myself and Joe Kershaw that run the physiology lab here at SMRU to sort of think a little bit broader and to sort of, yeah, sort of go beyond just the general ecology and physiology into sort of thinking beyond marine mammals into, into other animals, but also potentially into in the future into humans. So I have to say, familiarizing myself with the EvMed, I have to say, as an ecologist, this is quite a daunting prospect of speaking to folks from your background. But I do hope my sort of relatively limited scope will be made up for by the I'm biased, but the sort of incredible animals that we study. And really the sort of branch between evolution and ecology. I know we hear in ecology a lot that ecology is nothing without genetics, but I think even Darwin himself would flip that back to say that I think genetics is nothing without ecology, right? So it's how has evolution been driven to allow certain specific types of behaviors, and I think this combination of evolution, physiology, and behavior ecology, I think seals are probably one of the best animals, on the planet to study. So the vast majority of my research, 95% of it, is focused on the what we call seals, or well, pinnipeds. Pinnae meaning flipper, and then ped, of course, meaning foot. And then sort of, I guess, more normal parlance is what we call the seals. But before jumping into that, I just a little bit of sort of brief ecological background, as it's quite important to well go for the rest of the talk. So there's really four types of pinniped. The first being on the top left of the screen here, the sea lions. They're sort of the quite beautiful looking, quite charismatic animals. Quite distinctive is their external ears, but also looking at their back flippers is their ability to effectively rotate them around and pull them in under to act a little bit like hind limbs, which gives them the ability to sort of run on land. They're quite quick, also quite large animals. And quite similar to them are the fur seals. They're a little less cosmopolitan, tend to live a little bit more remote, a lot smaller than sea lions, but also have the external ears and the ability to pull the back flippers in underneath themselves, and so are quite mobile on land. And then the third type are, I think most people will know, the walrus. So those are three pinnipeds. They're closest sort of, they're a little bit evolutionary clustered with their sort of I guess their sort of ancestor being a little bit more closely related to the Ursus, to the bears. And the fourth kind, which is the kind that I'm going to talk about, are what we call the true seals, or the phocid seals. Phocid, basically from the Latin phoca, for seal. So these are the vast majority of my time are focused on these animals, and as I hope I can sort of get you to agree or believe, is that these animals are spectacularly well evolved. They are the pound for pound the best diving animals. So the phocid seals range in size from anything like 40 kilos from the small Sima ring seal up to about 3,000 plus. I put a plus because it's basically logistical limitations in being able to measure the mass of these animals. They sort of span polar to temperate waters, so generally cooler waters to cold waters, but they differ in their evolutionary ancestor more so from the other three pinnipeds. Their evolutionary ancestor, rather than being bear-like, is more like a semi-aquatic otter-like carnivore, Pugila darwini. And in my opinion, they're a little bit further along the evolutionary transition from a terrestrial mammal through a semi-aquatic animal, really to being a true marine mammal. And when I say that, I mean that they spend the vast majority of their life in water. They are bound to land to molt, to lose their fur, and also to birth their pups. But other than that, these are really fully marine animals. Now, phocids, when I say marine animal, like I mean they really are true ocean travelers. And just to give you a bit of context, can I just check? Can you see my mouse? I'm using it as a pointer. Yeah, great. So this gray lump here, this is the southern coast of Australia. This is Tasmania, and this is the southern tip of New Zealand, and this is the Antarctic continent. And these green dots here are tracks from southern elephant seals from the Australian island called Macquarie. And the blue here in amongst this blob is the island of Kerguelen, a French island, basically. And due north of there would be the southern tip of India. And so the sort of transit distance here from Kerguelen down to the Antarctic Peninsula is about 1,200 miles. So I put that online, and basically that means from Omaha, Nebraska to Tampa, Florida, right, is a straight line. But these animals are not making straight lines. I mean, they're making transits over weeks down to get to the continent, and making long transits around the ice sheet and then back again. But of course, beyond that is that they're not just making these sort of 2D horizontal transitions. These are all interspersed by dives, anywhere down to much deeper than 1,500 meters. These animals will go to 2,000, 2,200, even 2,500 meters. And I mean constantly. So it's making, a trip from Omaha Nebraska to Tampa, Florida, but continually going up 2,000 meters and down 2,000 meters. But it's not so much how deep they dive or how long they dive or how far they go that to me is important in the evolution of the animal. I mean, almost all of their life's activities, foraging for fish, finding fish, making transit to where they want to get to, a lot of their interactions with individuals. I mean, even some evidence for that's where copulation occurs, happens in the water. So the most important thing for them is not how far they go, how deep they can dive. I mean, I think that's just where you are, where you evolve. If your food is only 2 meters under the water, it doesn't matter if you can't dive 2,000 meters. What really matters is the percentage of time that you can spend underwater. That's the key. That's been one of the key evolutionary drives is to maximize their ability to do that. So we put this in context, if this is our percentage time submerged, you've got, really on the very edges of what we would call a semi-aquatic animal is the American mink. They're about 1 to 1.5% of their time underwater. The Eurasian beaver, which I think most people would, when you think of a beaver, you think semi-aquatic, is only about 4 to 5% of their time underwater. Whereas if we look at a real marine mammal, a sea otter, that's about 52% of their time underwater. And then the New Zealand sea lion, a real pinniped, that's about, again, 50 to 53% of their time underwater. Some work we did on free diving humans, an amazing group of all female divers from the island of Jeju in Korea, the Henyao, and they have about 3,000 years of provenance in diving. And so we instrumented them with some of our tags just to basically measure their fine scale diving behavior. And we found that they were spending about 57% of their time underwater. And some of these women were diving for sort of 8 to 9 hours a day. So a huge amount of time underwater. Remarkably, the youngest of the Henyao we instrumented was 69 and the eldest was 82. So these were very much in what would, I guess, for they would call the twilight years, still remarkable dive capacity. But then if we look at a gray seal, just a very normal sized true seal, I mean, 90% of their time underwater. So I think this is a fundamental shift from being a terrestrial animal to a terrestrial animal that will dabble in the water, something like a jaguar, to a semi-aquatic animal like a beaver, to a real marine mammal like an otter or sea lion, to being the most accomplished pound for pound best diving animal. So again, I've sort of phrased it as seals are diving animals, but I sort of hope you'll humor me here is that it's a little bit of a semantics or a little bit of philosophical gymnastics, but to me it's really, really key is that seals are not animals that live at depth and occasionally returning to the surface to breathe. Sorry, animals are animals that live at depth and only occasionally return to the surface to breathe. They're not surface dwellers, which only occasionally leave the surface to breathe, right? These are animals that live their life underwater, but are still fundamentally constrained by the same resources as we are. And the key one being is this dissociation from available oxygen. So as such, they live a life dictated by the oxygen economy, where access to this life-sustaining resource is finite. And so the evolutionary pathway has adapted autonomic responses to prioritize oxygen economy. And that's sort of what I would like to talk about is that there seems to be evidence and growing evidence that just fundamental, let's call them mammalian, I think they go further back than that, fundamental autonomic responses that have been rewired and have had to be rewired in order to allow these animals to be able to spend as much time under the water as they do. And as I sort of hope what are the potential learnings from seals that may be helpful in understanding autonomic function and nature's incredible ability to select things to work optimally and what we might learn from that to understand potentially human illnesses in the future. So I'd like to start off with cardiac function in seals, something I find quite remarkable. So this is some heart rate data from free-ranging grey seals collected back in the 90s by some of my colleagues here at SMRU. So these are adult female grey seals out off the Hebrides, the North Outer Hebrides in Scotland. So these are fully free animals diving of their own volition. They basically had an EKG that then was converting the EKG signal into sound and transmitting that sound. So basically following the animals around in a yacht with a hydrophone, just recording, like analog recording the heart rate signals sound coming back. And so a normal sort of inter-dive interval or heart rate between dives is somewhere between about 100 to 120 beats per minute. And this arrow here shows the onset of the dive. And so at the onset of the dive, you can see your heart rate very rapidly drops down to, in this case, 4 beats per minute. And then it holds at that 4 beats per minute pretty well. You get a few sort of like slight changes in heart rate, which subsequently we've seen as basically sort of very brief periodic increases in muscle perfusion, just to get a little bit of oxygenated blood back in there to re-oxygenate the myoglobin and then shutting back down again. And then at the end of the dive, the heart rate spikes back up to the 120 beats per minute range. So key from this is what I want you to take is that whenever they dive, heart rate drops, it stays at that level pretty much all the way through the dive, uh, independent of depth, fairly independent of exercise, and what the animal is doing. So it's just pretty much, we think of it as a sort of three sides of a rectangle. It shoots down, it stays, and it comes back up again. Now in this plot, this is again heart rate on our y-axis and dive duration on the x-axis. And these top triangles here are when the animal's at the surface. So I don't want you to focus on them. I want you to focus on the shape here of the heart rate during a dive. And what's fairly clear is that as dive duration gets longer, the heart rate gets lower. So this means, again, to reflect on the previous slide, is that from a dive is triggered, when the heart rate drops, it drops to the level that it's going to stay at for the dive, and it stays there until the animal comes to the surface. So this was some of the initial sort of first bits of evidence that indicate that there's some cognitive physiological matching between the magnitude of the dive response, so how much the animals are effectively shutting down, having a regional ischemia or regional reductions in perfusion to match the reduction in the rate of oxygen consumption rate to match the anticipated challenge of the dive. So again, and not just are they sort of effectively controlling how much, for want of a better description, dialing down the rate at which they're consuming oxygen to match how long they dive. It also seems that they've sort of, for want of a better word, hijack the ability to switch this on and off. So obviously for us, the dive response is trigeminally mediated. So if you hold your breath, and put your face into water, as long as that water is not too cold, you will get a nice dive response. You'll have a fairly moderate reduction in heart rate for a while, and then you tend to then get an elevation in blood pressure, and then heart rate starts to trickle back up again. These animals can switch it on with no external stimuli. So what we found in animals was one, we can train them to drop their heart rate on a command, something we sort of captured in terms of behavioral training, captured accidentally. But we've now got lots of data that show that we know that the animals at the surface breathing, respirating at a very constant rate of about 30 breaths per minute, face clear of the water in a nice sort of small respiratory dome that I'll show later. So there's no water lapping on the face or cold air hitting the face. And we know that they can just drop the heart rate whenever they want to do. So not just controlling the magnitude, but when this extreme autonomic response is triggered and also switched off again. We know they'll switch it off. They don't wait to the surface to reverse things. They'll switch it on depending on the dive, with a few seconds to many, many seconds before they surface. Something else that we also seen was not just heart rate, but of course, heart rate and peripheral perfusion, peripheral restriction, and restriction within the arterial tree and venous return are all very closely matched to maintain blood pressure. So what we find is looking at peripheral circulation optically with near-infrared spectroscopy while the animals are diving. We find that effectively the animals are setting the dive response off, increasing peripheral restriction, from effectively moving blood from the periphery back towards the core way in advance of diving. So this top plot A here, this is concentration. So basically the amount of, for the green line, the amount of hemoglobin. So if that goes up, there's more blood being measured. If it goes down, less blood. And each of these, this x-axis here is time in minutes. And each of these blue bars is a single dive. And these white bars in between are the surface intervals. So roughly comes out pretty close to 90% of the time underwater. So if we zoom in on one of these dives here in this blue box and look at figure B, th- this figure starts at the beginning of a dive. And so across the dive, the descent phase, inactive at the bottom of a dive, and then turning around to come back to the surface, we see a fairly relatively monotonic decline in the amount of blood in the periphery. So they don't just switch it off and sit there. There's like a sort of continual increase in the magnitude of the dive response. But what we find is then when they get to the surface, as we'd expect, they're sort of reversing that, and the peripheral perfusion is increasing again. So it's like a post exercise hyperemia, as we sort of expect to see. What we find is that each dive, about halfway through the surface interval, this is when the animals will start to trigger the dive, the increase, the dive response, the increase in peripheral restriction. So it didn't matter if that surface interval was 5 seconds or if that surface interval was 50 seconds. It was very close to 55% of the way through the surface interval. Again, with no external stimuli, obvious external stimuli, they're setting the response off. So it seems there's certainly some sophisticated connection between cognition, the animal's decision making, and how that's being realized in terms of the physiological changes that generally we would assume as being entirely autonomic. It seems it's not entirely autonomic. Now, that control of heart rate is obviously essential to the animals being able to dive, not just for a long time, but matching the anticipated challenge of the dive to how much or the magnitude of that response. So on land, seals demonstrate the expected cardiovascular responses to startling. So if a seal is on land and it gets a startle, it shows, just as you'd expect, heart rate will shoot away up, peripheral perfusion will increase, and generally there's an increase in ventilation rate as well. Now, as soon as the animal dive, as soon as they are holding their breath, that response is either absent or it's inverted. So there's sort of circumstantial or sort of little bits of evidence from sort of like little accidents that have happened, not fatal accidents, but just sort of where animals have anticipated the end of a dive and then that's not been realized. That startle response creates a reduction in heart rate and likely a reduction in peripheral perfusion as well. So we wanted to look into this because I think if these animals have control over their heart rate and they're able to or dive response, and whether that's vascularly mediated or cardiac mediated, I would probably put my eggs in the basket of vascular mediated, is that these animals cannot afford an elevation in heart rate if they were to get a scare. So if we had an animal that was at 2,000 meters, I mean, some of these animals will run arterial PaO2 down to 14 millimeters of mercury, so incredibly low. They don't really have a huge amount of bandwidth to exceed their dive duration. So the last thing they can afford is that, say, they encounter a predator, and they're, anywhere 10 minutes away from the surface and access to ambient air. They certainly can't afford to then massively increase their rate of oxygen consumption. So what we initially tested was we wanted to see whether or not the sort of the cyanoatrial node is responsive when the animals are holding their breath to beta blockade. So we did an initial bench test where we delivered a beta blocker, direct infusion, venous esmolol. So we gave a mass specific dose, a second mass specific dose, a third, a fourth mass specific dose, and then we give a quadruple dose to the animal. So the decay rate we expect to be about 10 to 15 minutes. So we'd wait 20 minutes between doses, but we give effectively 1, 2, 3, 4, and then a quadruple dose, and absolutely no effect on heart rate, no effect on respiration rate. It didn't affect either the animal's ability to, when it's breathing, to bring the heart rate up, and then when it was holding its breath, to take heart rate down. We may as well have injected saline into the animal. So now what we want to bench test is the delivery of adrenaline and whether or not that has no effect on heart rate. And our initial tests, again, it's like injecting saline. When the animals are holding their breath, adrenaline has no effect on heart rate. As soon as they start to breathe again, then we get the normal response. So adrenaline would give a normal effect. Heart rate skyrockets, peripheral perfusion increases. However, if we test the delivery of phenylephrine or atropine, so vaguely mediated pharmacological agents, we get the expected very nice bradycardia and very nice tachycardia. But whenever we deliver the adrenaline, whenever the animals hold their breath, what we see is this gradual presentation of grade 1 initially transitioning into grade 3 atrioventricular blocks. So we think the animals basically have trigeminally mediated ability to sort of create atrioventricular blocks to sort of shift the pacemaker of the heart down to the atrioventricular node, which teleologically makes sense. As I say, I don't think these animals have a lot of bandwidth to get a scare underwater and for that to rocket heart rate. So this seems potentially an adaptation that we want to explore. And we'll do that properly in January with the animals we have in our facility. With a series of experiments, we'll deliver adrenaline underwater, we'll deliver adrenaline remotely in the dry, and then have the animals, if we get that nice elevation in heart rate, have the animals then get into water and see whether or not they can decay the effect. And then we're also interested in trying to develop some cardiac organoids with a colleague that recently started in St Andrews, who basically has been using fibroblasts from different species to create artificial leather for companies such as like Hermes and I forget, not Rolls-Royce, Bentley. So yeah, they've developed cardiac organoids for different species. So we would quite like to develop cardiac organoids and that have, you know, a sort of pulsation and see whether or not, yeah, it's something that's neurally mediated or whether it's something sophisticated within the organs themselves. So I think the control of the cardiovascular system and this sort of effective this relationship between cognitive decision making and bulbar responses, I think, is very key to their economy of oxygen. But of course, you can't talk about the economy of oxygen without also talking about the economy of CO2. And I think CO2 is as heavily weighted in their evolutionary adaptation as oxygen is. So I have to do a little bit of work on freedivers and have done in the past. And one of the things that really didn't sit right with me was going to the competitions, the world championships, where you have really the best of the best there, competitors like Alexei Molchanov. Um, it was quite concerning. I mean, to them, totally fine. It's part of their sport. But to me, remarkable was the number of people that lose consciousness on their way back to the surface. Also, the amount of pulmonary edema. Um, and as an ecologist watching that, you very quickly realize that in the transition from terrestrial to semi-aquatic animal to full marine mammal, you don't have any bandwidth to make that mistake even once. I mean, if you know, especially seals that, you know, they tend to exhale before they dive, so they're negatively buoyant, quite shallow. If you lose consciousness even once at, you know, even a few meters deep, these animals, that's it. You're out, your fitness is zero. You're out of the population. You're not going to breed. So it seemed to me that these animals have to be able to have some idea about not just where their oxygen levels are, but being able to do away with, I guess, the really pronounced autonomic, but also cognitive manifestations of elevated CO2 that we experience. So my hypothesis was, excuse me, our hypothesis here at St Andrews was that these animals have some way of knowing where in the grand scale of, you know, 100% saturation to zero saturation they are, and don't really care too much about CO2 for a number of reasons, as I'll get into. So we have a short-term animal facility here at the Sea Mammal Research Unit, which is quite globally quite unique in that we can bring harbor or gray seals from the wild, our local populations, into our facility for a while, and then we release those individuals back into the wild again. And for the most part, we usually instrument them just to make sure they integrate back into seal society normally and behave normally. We've now had 100, we're on our 133rd animal, and yeah, we know these animals go back and you know live sort of normal lives and end up being breeders as well. So and we work under quite a strict licensing system. But within our facility, we have a large pool. It's a 40-meter pool, but we tend to break off the last 10 for where the animals reside into satellite pools. So we have a 30-meter experimental pool. And in that pool, we basically have a very simple maze where the animals have access to a small breathing chamber. And then they have to make a dive, sort of 30 meters around this lane divider and 30 meters back to a feeder where they can spend as long as they want at the feeder consuming fish and then transit 60 meters all the way back again to take a breath. So it's a sort of 2D simulated diving setup. And then when the animals are in the breathing chamber, that gives us the ability to control the concentrations of the inhaled gases that they're breathing. So we would either have the animals breathing ambient air, or hypercapnic air, where we took CO2 from 0.04% up to 8%. We actually went higher, but we came back to 8% because we'd written in our license that we would only give them a maximum of 10%. Then, of course, on the very first breath, when they get to the surface and exhale, you exceed 10%. So we reined that back to 8%. We also give them hyperoxic air. So we're keeping CO2 at 0.04 and take oxygen concentration up to 50%, or hypoxic, and we took concentration of oxygen down to 11%. So in terms of what our facility actually looks like, this is the pool itself. On the right here, this is where the breathing chamber is fitted to. And then we cover the surface in these surface panels. So the animal has to make that underwater transit to the feeder here. And the bottom here is this sort of big, it's just a fish conveyor belt, really, with a set prey-delivery rate. Then the animals swim back again. And so all we do is we have a gate here where the animals are residing with their, we usually have 6 at a time. We just open this gate. So everything that the animals do in terms of their participation in it, what they do when they're in there is all volitional. So if the animal doesn't want to be involved in it, the animal isn't involved. So this is very much all on their terms, which is what we want. We want them to effectively, we set them a problem and we want to see how they solve it. But working with green animals, as we call them, these are animals that come in from the wild. We do have to kind of temper our expectations and how much that we can achieve in a 12-month period. But the diving experiments, they seem to love so much so that when we open this gate, it's very much akin to anybody that has a dog. It's like lifting the dog's lead or lifting the dog's collar to go for a walk. It's like these animals, getting them in isn't the problem. It's one thing that the animals wanting to get out again is usually our issue. So just to be clear that everything is very much on their terms. We don't force the animals to dive. We don't force them underwater. Indeed, if the animals don't want to be in there, they can exit. That's part of our license agreement, is that the animals can leave if they want to. So this is one of our animals. This is Oshin in the breathing chamber where we had the mixed gas. And again, in the training process, if the animals don't want to be in here, they don't. But the animals very rapidly choose to, rather than breathing at the surface, to breathe in the chamber. Even though they're marine mammal, remarkably, seals hate the rain. They despise the rain. They won't come onto land if it's raining, and they really don't like the wind very much. And there's a lot of combined rain and wind in Scotland, which works to our benefit. We're actually training animals at the minute to do this, and it's so easy to get them in there. So I think a lot of people see this and sort of think, Jesus, you must be really forcing the animals in there. But I say, really, everything we do, we don't really train. We allow the animals to sort of discover their way. But breathing somewhere, swimming to get food is the most natural thing for a seal. So they really take to it very, very quickly and easily. And then when they're in there, we just have a series of cameras that we can watch what the animals are doing. So we can track dive durations, dive behavior, and then we have the ability to put instruments on the animals as well. So in terms of putting instruments on the animals, one of the really difficult things in I guess working with any large mobile mammal, particularly animals that live underwater, because you can't easily get access to them if you need to handle them, to take a blood sample. We also don't like to do that. It sort of breaks our trust relationship we have with the animals quite quickly. So to get around that for this study, we designed and fabricated in-house a remote animal-borne blood sampler, which we call the Mosquito. It's 2 S's because our colleagues called Simon Moss, so it's not a spelling mistake. It was quite a simple system, all made in-house. It's basically a 3D nylon printed housing with a Delrin machine in-house cap connected to a cannula that we put into the extradural vein. This is a large vein, quite large, probably about, thicker than your thumb in animals, these sort of 40 to 50 kilo animals. Large valveless vein with anterior and retrograde flow in it. So it's very easy to pass a cannula in and pass it quite far up into the vessel. Then that sort of houses just a battery pack, a small stepper motor, a little 3D printed cradle that makes up a peristaltic pump, some solenoid valves, and our syringes connected to a lure lock lid. And basically what that allows us to do is to draw blood from the animal, but basically draw to waste to clear the cannula, to take blood samples and then push heparin back down the line to maintain patency. So this basically allowed us we had to manually handle the animal once to drug it, to place the cannula. And from that point onwards, we were able to remotely sample blood, but also able to remotely sedate the animal with butorphanol and methamphetamine, which meant in terms of our 3 Rs and improving the animal experience and the quality of the science, we didn't have to handle our animals to get blood, and we didn't have to repeatedly handle our animals. So we were able to keep a very good trust relationship, which means we can do the experiments quicker, because if the animal won't trust you, it's difficult to get them to move pools, which means we can reduce the time that we keep the animals in the facility. And one of the key reasons I wanted to sample blood is, of course, you can't really talk about O2 and CO2 without talking about pH. So we ended up collecting about 500 dives across our 6 animals. And then we would take a blood sample at the end of a diving trial where the animal would make 5, 6, 7 dives to the feeder, breathing one type of oxygen or so one type of gas condition. This baseline blood sample, this is what we would take when an animal's lying on the side of the pool asleep. We would just take a blood sample to get what we would call probably the most reflective baseline you'll get in a marine mammal. And then whether the animal was diving in ambient, hypercapnic, hyperoxic, or hypoxic gas conditions, although there's a general trend towards very mild, well, acidosis, none of these were statistically significant. So these animals can make repeated dives, breathing hyperoxic air, hypoxic air, breathing 8% CO2. They do not change pH. They're incredibly gifted at regulating and balancing pH, which is something I'll get into after. So in terms of the diving behavior, what we found is this is dive duration on the y-axis. On our x-axis, this is our gas conditions, is that although there's a general downward trend for CO2, it was not statistically significant. So the animals were not making diving decisions on how long they stay underwater based on CO2. Instead, they were diving longer when there was more oxygen available and diving shorter when there was less oxygen available to them. However, at the surface intervals, so this is the time they spend at the surface between dives, CO2 is the dominant driver. So although it's not affecting the decisions they make when they dive, it's absolutely affecting their decisions they make when they're at the surface. They spend a lot longer at the surface when they're breathing high CO2 and longer when they're breathing hypoxic air, but nowhere near the effect of CO2. If we look at the animal's respiration rate, quite interesting. So on our x-axis here, this is our 6 animals, and on our y-axis, that's the breaths per minute. And then each of these color bars is a different gas condition for each animal. But the sort of big takeaway there is that neither O2 or CO2 is affecting these animals' respiration rate. It's almost a respiration rate it's basically set within an animal, roughly somewhere between 28 to sort of 40 breaths per minute. So independent of breathing CO2 for an hour, 8% CO2 for an hour, or hypoxic air or hypercapnic air or hyperoxic air for an hour, respiration rate really doesn't change. Maybe a marginal change in tidal volume. That's something our student Anne is looking into, but respiration rate doesn't change. If we look at peripheral perfusion, it sort of fits a little bit more with what we'd expect in a mammal. No change in peripheral perfusion, the magnitude of reduction in peripheral perfusion when they dive, but when they're at the surface, there's an increase compared to ambient and peripheral perfusion with hypercapnia. So I guess what we'd expect, but fairly attenuated. And again, an increase in peripheral perfusion slightly with hypoxic air and a reduction when they're breathing hyperoxic air. So that fits a little bit more with what we'd expect in a mammal, but not so much the respiration rate. Or I think in general, the sort of diving decisions and decision making based around O2 and CO2. So again, you can't really talk about the economy of O2 without CO2, and you can't talk about the economy of CO2 without pH. But as I sort of demonstrated previously, we couldn't get these animals to change pH. So we have no idea what the effect of pH is on behavior, nor on sort of just fundamental autonomic responses such as respiration rate or peripheral perfusion and heart rate. So what we're doing recently is just bench testing. This is experiments we'll start in January, is basically pharmacologically affecting pH. So we wanted to induce first acidosis, and we did that with the inhibition of carbonic anhydrase through the delivery of acetazolamide, or a lot of people maybe know it better as Diamox, used for glaucoma and altitude sickness. So we brought an animal in on the bench. This is an animal under sedation. And at 0 hours here, this is our delivery of the acetazolamide. So initially, I'd like to get you to look at PCO2. So we deliver at 0 hour. PCO2 was quite high because the animals under sedation, like when they're sleeping, they hold their breath. So after an hour and a half, PCO2 was higher again. Three hours later, the animal is then up and awake and moving around. PCO2 was low. It sort of jumped back up again. 6 hours later, again, probably a like of another handling and sedation event. And then on the last 9 hours, it was back down sort of around 70, so still quite high. What was quite interesting though is what we wanted to achieve is if we look at blood pH. Blood pH again was quite acidic when we started. This normally, as I showed before, should be 7.3, but this got all the way down to 6.84. So this animal was very, very acidic and was very acidic for hours. No change in ventilation rate, no change in heart rate. That animal happily responding to training, happily playing with the conspecifics, eating. Again, outwards it looked like he'd deliver saline, but internally, yeah, absolutely, this animal's very, very acidic, but not having the sort of behavioral or autonomic responses that we would expect. What we also try to do is we try to induce alkalosis. And to do that, we just delivered about 70 mil of 8.4% sodium bicarbonate. So this -20, this is our baseline measurement. So, I mean, venous bicarbonate, there's about 20 millimole per liter. Then the red band is the start of the infusion, and the other red band is the end of the infusion. And so you can see delivery of bicarbonate, you get this spike in bicarbonate as we would expect. And we'd hoped we would sort of maintain high bicarbonate levels for maybe 4 to 6 to 8 hours. On our next sample, all the bicarbonate was gone. So this was, 10 minutes after the end of the 70 mil infusion, bicarbonate is back below baseline levels. So in real time, quite hard to digest, what's going on. And we serial sample, so we take two to four blood samples in each time point to sort of just correct for any potential spurious results. If we look at their PCO2 went as high, so this bicarbonate is rapidly getting converted into CO2. So PCO2 went as high as our system could measure, 130 millimeters of mercury. It was higher than that, but basically we just started getting non-readable results. So we actually had to take the cap off the blood tube for a few seconds and put it back on to let some CO2 out just to get the system to make measurements. So we don't really know how high CO2 went. And then started decay again after about 20 minutes back down to sort of lower levels, but still quite high. Again, probably the effect of sedation. Blood pH, as you'd expect with CO2, the initial delivery of bicarbonate, we got some slight alkalosis, and then we got real acidosis. The animal was at 6.75. No change in respiration rate, no change in heart rate, no obvious changes in behavior. The animal wasn't becoming acutely responsive to anything. Again, it felt like we had delivered saline. So if we piece those sort of together with the O2, the CO2, and the pH, we kind of get this collection, one for a better description of phenotype, which is that says show no aversion to significant hypercapnia. Perception to CO2 seems highly attenuated, but by no means extinguished. It does affect the surface intervals. Hypercapnia has no effect on respiration rate, no effect on the magnitude, it affects reperfusion, the magnitude of peripheral perfusion. And similarly, extreme acidosis does not affect respiration rate, heart rate, or peripheral perfusion, nor does it generate aversive responses. So this sort of leads us to obviously query, well, how is that achievable? And sort of a chance conversation I had with Professor Jack Feldman from UCLA, who I think was quite influential in the discovery of both the retrotrapezoid nucleus and the pre-Botzinger region of the ventral brainstem, was, well, how can animals not care too much about acidosis or extreme hypercapnia? And he sort of pointed initially towards the sort of field of this could be function in the retrotrapezoid nucleus in the brainstem, sort of key chemosensor regions of the brainstem, with a lot of projections to this pre-Botzinger region that controls respiration. But he put me in contact with Professor Debbie Wiesmeyer, who's a clinician that works on a specific majority on a condition called CCHS, or chronic central hypoventilation syndrome, where children are born without ventilatory drive. So they're unresponsive to CO2 and unresponsive to acidosis, and so usually have to have a tracheotomy and be mechanically ventilated, almost always during sleep, sometimes during awake and sleep. And in our sort of discussions with her, she basically described the phenotype very well of the seal, so much so that one of her patients, tracheotomy, so had never swam, really had survived to teenage, still alive at teenage years, and wanted to swim. So she basically blocked up her tracheotomy and got in a pool, but had never been in a pool and didn't realize she couldn't swim. And she sank to the bottom of the pool. She described it to Debbie as that when she was on the bottom of the pool, she was looking up and she could see her brother and was completely fine. No aversion, no desire to breathe, was just looking up and was quite calm. Was like, my brother's eventually going to see that I'm down here and it's going to come and get me out. And that to me very much has to be the way seals' lives are. I don't believe that they spend, 90% of their life with chronic anxiety because of high CO2, high ventilatory drive. So now we sort of looking towards the effect of this gene that causes CCHS and other conditions called FOX2B, that has sort of quite a clear, distinct mutation that generates this phenotypic response. And so now, along with Jack and Debbie, but very, very closely with Jason, and Jason very much got us thinking about this and how to think about this in a really exciting way, was chatted to Jason. And Jason was like, well, look, this is much more beyond the sale thing. Like, we need to go and look at this more broadly. So let's look at the across the mammalian tree. So we know that the mutation in particularly this alanine repeat section of FOX2B, so the normal alanine repeat section towards the C terminal of the gene is normally 20 alanine repeats. And if we look at that across the vast majority of mammals, just a few here, cows, horses, camels, hares, jackals, rats, chimpanzees, vast majority of animals have the normal 20 alanine repeat. Now, when that alanine repeat gets longer, that's what leads to, can be 23, 24, 25, anything up to 33, generates this congenital central hypoventilation syndrome. And we see some animals, remarkable to me is the platypus and the echidna. So they're the monitoring mammals. They're the mammals that lay eggs. And some of the hibernating animals like the spear-nosed bat. But if we look at deletions, which also sort of linked to these apparent life-threatening events, which is lack of response, apnea, not breathing, and it's complicated, but also potentially related in sudden infant death syndrome. If we look at that, we see a whole variety and almost a who's who of diving animals, or animals that are routinely exposed to chronic or intermittent hypercapnia or hypoxia. The walrus is in there, our seals, river otters, other seals, the manatee, the otters, and the moles, the real who's who of hypoxic hypercapnic tolerant animals. And so we're starting to develop a hypothesis that this gene may be central, this highly conserved gene. Now, this gene is in sponges. It's been highly conserved across the evolutionary time, from the Cambrian period, 350 million years ago, into the reptiles and then into the birds and mammals. We think that this gene, we want to explore that hypothesis along not just the phenotypic responses, but also looking at, at the levels of the cell and of the tissue, whether or not actually what is quite detrimental, very detrimental in humans, has actually been essential and strongly positively selected for in animals, a whole variety of animals, to allow them to be diving animals or animals that routinely experience hypercapnia. So that's something that, along with Jason and Gillian Wissey and Jason's group, we're starting to pursue. One of our nice first really easy hypothesis test is I accidentally stumbled into helping people with analysis of NIRS data who had replicated our SEAL study in humans, and 8 of their 40 participants would breathe 8% CO2 and perform exercise, 200-watt output exercise on a static bike, had no idea for 40 minutes that they were breathing 8% CO2. So we've now got blood samples from them. And I think if the FOX2B alanine repeat section mirrors what we see, especially on the lower half there, is that these are subclinical subclinical FOX2B mediated attenuated responses to CO2. They didn't have any respiratory response. They didn't have any cardiac response. They also went acidic. They had no responses to the acidosis. Is that I think that this gene and selection on this gene might have potentially quite a improved broad evolutionary understanding of the drivers of evolution of a whole suite of diving animals. And if that is the case, then I think we have a whole suite of ranges of the mutation of FOX2B that may present a whole suite of phenotypes that could, as I'm led to understand from Debbie and from Jack Feldman, that could provide a whole suite of new models to understand both CCHS, but also these apparent life-threatening events with deletions in FOX2B. And so I want to leave in just something incredibly unscientific, something that jumped out to me, a little bit of Jungian synchronicity. So CCHS, until fairly recently, was named Undine's curse. And of course, that's negative connotations, the word curse in a condition. So it was changed to CCHS. But the story of Undine, the reason it was Undine's curse, was based on the story of Undine, a water nymph, and whose husband, cheated on her. And she cursed him that he could never sleep again because if he ever went to sleep again, he would die because he would never be able to breathe. But Undine is very much, the character Undine is very much taken from the Irish, the Celtic folklore of Selkies. So water women are the Skelta Ronta, the Skelta Ishka, the water women, which are seals that turn into women when they come to land. So I have a beautiful Jungian synchronicity where this Undine's curse, this condition, and potentially that seals may actually provide some better understanding into that condition and this sort of connection with folklore. It sort of definitely has quite a deep hook in me now that I really don't want to let it go. And I'm really, really looking forward to combining the stuff that we can do. You know, we can put seals into MRI and we can induce acidosis. We can do the diving experiments, but really working hand in hand with Jason and taking things down to a much more fundamental cellular function, tissue function, transcriptomics, proteomics, metabolomics, and when is this gene being switched on? Is it switched on and off? When is it transcribed? I think could, yeah, not just for understanding cells, but much broader in terms of mammalian, potentially avian and reptilian evolution, as well as these really unpleasant conditions in humans, which is very much outside my application, but I hope maybe this could snowball to that. So with that, there's really is a huge amount of people to thank, but I just really want to thank the core team here at SMRU because when we have the animals in the facility, I really mean we give up life. It's 7 days a week, long hours in the cold, caring for the animals. And also, again, to reiterate, a huge thanks for the invitation to talk. And I hope there's at least some of that in there that's sort of interesting or can stimulate some thought. And so with that, thank you very much. And I'd be happy to, yeah, take any questions and sort of discuss things. Thanks very much.
Yeah, Chris, that was awesome. Thanks so much. Appreciate it.
Pleasure.
Lots of claps. Folks, if you have questions, please, feel free to raise your hand on the Zoom or unmute, throw it in the chat, whatever way you want to do it. And I'll sort of I guess I'll kick us off. I mean, I have lots of comments, questions. I was just curious what you think is happening in cetaceans, um, because I see that
Yeah, yeah.
so anyway, if you have
So
thoughts on that, I'd love to just hear
yeah, and
philosophy of it.
yeah. Well, and again, it was I had this great hypothesis, and then you threw the complete curveball into it, which is all like the most elite diving animals don't have it, which are the cetaceans, and that again it was a great impetus to sort of dig a bit deeper and think about this more, and really just to think about it. So those there's if we think of diving mammals, excuse me, you've got four main groups. You've got the carnivores, right? So you've got the polar bear, the seals, and the diving mustelids, the otters, and others like mink. Then you've got rodentia. You've got the diving semi-aquatic rodents. So beaver, of course, is a classic, but there's a huge number globally of diving rodents. Then you've got the Sirenians, which are in Aphrotheria, and there seems to be selection on there, and I think there's selection in Tenrec as well, a little Madagascan, small, looks like it should be a rodent, but it's not, a little diver. The one where you don't is Artiodactyla, which are the cetaceans, and I think evolutionarily, certainly within Rodentia and Carnivora, these are all search and pursuit animals. So effectively they have limited bandwidth to dictate the time that they spend underwater if they're limited by the amount of time they have to spend at the surface. So they got to search, find their prey, and then they got to be able to pursue that prey. Fair enough, it's insectivores, and then I mean large carnivores. I think within Artiodactylum, because if you think of cetaceans as effectively, I mean, they've still got lots of stomachs. They're basically a really good cow. I think evolutionary, they have come from things that are quite passive. If you just have to forage on the bottom or forage at the surface and graze on vegetative matter, you really can't. And if you're big as well and you have limited need to avoid predators, you don't have to spend, you can, if I want to spend 50% of my time underwater, you know where your prey is, it's at a set depth, and you can just go to the surface and go to depth and come back to the surface. So I feel like there's not a huge, maybe the same ancestral evolutionary pressure to tolerate CO2. And I think speaking to he's also, I should say, very influential in our thinking and our work with us is Andreas Fellman, is that, yeah, he has exposed animals to dolphins, to bottlenose dolphins to high CO2 just by a pump not running high enough. So we a little bit of build up in CO2, and they absolutely do hyperventilate and seem to show a lack of comfort with it. So I
Right.
think that what's initially nice, well, when I came up with the cetaceans, I was quite concerned, but actually that there's no obvious selection in Fox2b, that they seem to have aversion and respiratory responses, where what is available within the rodents and the at least in seals is that yeah, there's these attenuated cognitive or behavioral responses to hypercapnia in particular. At least we've demonstrated initially to acidosis, extreme acidosis, I would say as well, um, and the lack of ventilatory responses. I think it's very early, of course, but I think there's a little bit of enough of a picture immersion that I think it's worth pursuing whether or not Fox2b and expansions and contractions in the repeat section do underpin some of these phenotypes, and quite broad, potentially broad phenotypes.
Yeah, absolutely. Yeah, thank you. I want to get to Cynthia. Dr. Beal is asking two things. What's the metabolic rate change during a dive or with changes in CO2 and O2? And then what's the activity pattern underwater? Is it start and stop, or is it more smooth, and how do they evade predators? So a lot there, but maybe in a minute
Yeah.
or so.
So in general, the reduction of a very so I'll just initially take the gases out of it. The short answer is when we give them the gases, there was no change in metabolic rate, no statistically significant change in metabolic rate. But in general, a very extreme reduction in metabolic rate would be about 60 to 70%. And I think that sort of spans, from that 60 to 70% all the way back up to just a few percent. Again, depending on how long they anticipate the dive is going to be and how long they expect they're going to be underwater. In general, the reduction from non-diving metabolic rate that we would get is about 30%. So not, I mean, relatively big. It's much greater than I understand than we would have in sleep, but not 70%. But ambient, hypercapnic, hypoxic, and hyperoxic, there was no statistically significant changes in metabolic rate. In terms of the activity in our facility, it's just the transit, and then when they get to the feeder, they sit pretty still, and then they transit back again. In the
Mhm.
wild, there's a whole variety. They tend to just cruise about 1.3, 1.5 meters a second. And then once they reach negative buoyancy, they tend to just float. It's a little bit like a falling leaf. Sometimes they sort of turn. I mean, most of the center of gravity is quite low. They tend to then flop up the way and then float like a leaf. And then once they get to depth, then they sort of swim around,
Mhm.
looking for what they want. And then because they're negatively buoyant, they have to swim all the way back up again. But whenever our animals are going from land to somewhere to feed, it's pretty much they just swim, just nice,
Yeah.
constant. They'll only dive maybe, 3, 4, 5, maybe 10 meters until they get to where they want to and just cruise at that 1.3 meters a second.
Yeah, that's great. Thank you. I'll give one more question. We're right up against the hour, but
Oh, yeah.
I thought this was great. Like this person, Ned, I don't know if you want to chime in, but curious to know whether diving behavior and associated physiology are altered in pregnant females.
Yeah, definitely. Yeah. So behavioral wise, we can tell from our remotely, if we have tags on animals, we can tell they're pregnant. Their diving behavior, usually within about a month to 6 weeks before they give birth, gets very restricted. They don't dive. They still dive a lot, but their dive durations and their dive depths get quite small, quite obvious. She's also close to probably about 43% body fat, and she
Mhm.
has a 12-kilo pup in there. So I think she's got a lot of buoyancy to act against, but their diving becomes much more conservative. I think the females become much less risk averse. You see that in some of the Antarctic species where there's sheet ice, where the females, especially the adult females, won't go under the sheet ice. They tend to stand out with access to ambient air, whereas the juveniles and like juvenile females and adult males will go in under the ice. They're happy to take the risks. So yeah, certainly, yeah, being pregnant, absolutely, I think it affects their behavior, but I think a lot of that may be um, physiologically mediated. I think I don't
Yeah.
know off the top of my
Yeah.
head much about changes in hematocrit or changes in blood volume with pregnancy. I'd have to go and look at that because there certainly is that information. I just don't know off the top of my head what like changes in hematocrit or VO2 would be like.
Hmm. Yeah. Aaron's putting an interesting comment, maybe sort of the opposite of what you're experiencing in the seals, it sounds like, but yeah,
Hmm.
an interesting comment there as well. I know we're over time. I want to, respect that, but really, really great, Chris. Thanks so much for joining. And Charlie, I don't know if you want to close this off. Anything else to say?
Just to echo your comments, thank you so much, Chris. This was fascinating,
Pleasure.
really
Pleasure.
interesting. Thank you so much for sharing with our
Thanks
community.
very much for the invitation. I sincerely appreciate it. Thank you. Thank
All right.
you.
Thanks, everybody. Take care.
Bye, everyone.
Take
Bye.
care. Bye.